This method can help answer key questions in the vascular tissue engineering field, such as monitoring the dynamics of engineered vascular growth and remodeling during long-term culture. The main advantage of using optical coherence tomography, or OCT, is that it is a readily available, real-time, and non-destructive imaging strategy to characterize the structural features and remodeling process of engineered vessels. Demonstrating the procedure will also be Shangmin Liu and Wentao, technicians from my laboratory.
To begin this procedure, fabricate the PGA scaffold as described in the text protocol. Dip PGA scaffolds in 1 mol per liter sodium hydroxide for 1 minute to adjust the spatial structure of the mesh. Then, soak the scaffolds in tissue culture grade water 3 times for 2 minutes each. Each time, gently pat dry the scaffold with a tissue paper. Then, dry up the scaffolds in a hood with a blower for 1 hour. To assemble the bioreactor and the Y junction for OCT imaging, first soak the self-developed glass cylindrical bioreactor, PGA scaffolds, silicone tube, biocompatible tubes, connectors, stir bar, and equipment for assembly in a 95% ethanol tank for 2 hours. Pull the PGA scaffold through the side arms of the bioreactor connected to one side with a connector, as well as to another side with the Y junction used to deliver.